Hello class, today we would be looking at the neuronal physiology of how sight and hearing works. So these are including our special senses. So we are going to start with sight. When we see something, what happens is the light will enter our pupil, and that image that we see will be focused on the retina, causing it to be reduced and inverted. So what we see isn't actually already reduced or inverted. Our retina will have to do that. So let's draw a little diagram. So let's say this is our eye, right? And this is like our eyeball. You can think of your retina somewhere here, okay? This is so it will be induced, reverted on the retina. And then when that light enters, right, on the rods and cones, the rods and cones are found somewhere. So think of this as your eyeball, right? The rods and cones are somewhere here, somewhere in this space. And those rods and cones will basically go through chemical reaction that will then generate what we would refer to as degraded potential for our special senses, and that would be a receptor potential. So that will be here in step 2. The receptor potential occurs on these rods and cones. However, there will be no action potential. Then, after that, neurotransmitters from these rods and cones, so again, this is our eyeball, and if you zoom in, we have these rods and cones there. They will send neurotransmitters to a bipolar neuron. So, let's say this is not an accurate depiction of a rods and cones, but let's say this is your rods and cones, right? Not the most accurate looking, but let's say this is rods, cones. They will send those neurotransmitters, so NT, to a bipolar neuron. So it's going to send it to another neuron. That bipolar neuron looks like something like this. Again, not very accurate, but something like that. There's better diagrams out there, but generally that's what a bipolar neuron looks like. There's like 2 directions it's going to. So the neurotransmitters are sent to this bipolar neuron. This will then generate not a receptor potential, but just a regular graded potential. And when we generate a graded potential, we will then send neurotransmitters again. So same idea here, where it's sending from one piece to another piece, or one neuron to another neuron, or one special sense to so on, or one structure to another structure, is that it will send neurotransmitters to our ganglion cells. Now, ganglion cells, so let's say this is a ganglion cell. So it's like a giant bacteriophage. So it will send those neurotransmitters to this ganglion cell, and once it sends neurotransmitters, or our chemical messengers, this will generate an excitatory postsynaptic potential, and then eventually into an action potential. After this step, now that we have an action potential going on, as those neurotransmitters have been sent to the ganglion cells, we will then have those synapses going to the optic nerve. So again, this is our eyeball, and this is the optic nerve. So you could think of like your eyes, light going to the retina, processed on the rods and cones. Now it's being, now that we have an action potential going on thanks to the ganglion cell, we are synapsing here onto the optic nerve, composed of axons and ganglion cells, so many more of these. We will then send those signals to our optic tracts, and then eventually we will have those signals be sent to our visual cortex of the brain. So this would be our occipital lobe. So let's say this is a brain. Again, not the best looking diagram, but so brain stem, right? Generally cerebellum is down here, but so occipital lobe, so somewhere here. So the visual cortex, that's where the signal will eventually reach, obviously, in order to process this incoming image that we are seeing. And everything that we're seeing is just technically light, and that is what the occipital lobe will try to, eventually process as it goes through all these various steps. Okay, so let us look at hearing now. So now we understand a little bit on what is the physiological steps that takes place on the neurological level of sight. Now let us look at hearing. When we hear, hearing is again part of our special senses, not our general senses. The hair cell on the cochlear duct will be stimulated. So this will be inside our very ear, and if we remember in anatomy, the cochlear duct is somewhere like your eardrum, okay? There's those individual hair cells that will be stimulated because of vibrations, okay? Vibrations. So if you look over here, so this is our ear, for example, and then deep in our ear, we have these cochlear ducts, right? And they will be stimulated. Neurotransmitters will again be released to the associated neuron. An excitatory postsynaptic potential will occur, generating an action potential. Again, we have that, for example, here's one neuron. Here is not the best diagram, but let's say here's cell, right? And here's a neuron. Here's the neurotransmitters. It's being sent there. An excitatory postsynaptic potential will occur, having this action potential. Action potential will occur on the cochlear branch of cranial nerve 8. So this is a special nerve that is very important in sending signals to our auditory cortex, which is the part of our brain that is responsible for processing the things that we hear. So the auditory cortex, so again, this is our brain, right? Think of your cerebellum here and your brainstem. The auditory cortex is somewhere here. Okay? At the end of the day, all these signals will be sent to this part of the brain. Oh, sorry about that. And to be processed by the auditory cortex. Okay, I hope that gives you a little bit of understanding of what is the neurological physiology of our special senses such as hearing and sight. See you on the next video.